So welcome, everyone. I would like to welcome the speakers, the panelists too. So just let me introduce a bit EFFAB and FABRE-TP before giving the floor to our first speaker, the member of the European Parliament, Mr. Christensen. Yes, that's the EFFAB team, the EFFAB and FABRE-TP team. I wanted to introduce, we are 6 people now in EFFAB, and this webinar has been prepared by all of us. So that's I wanted to thank the team before starting. So EFFAB is the European Forum of Farm Animal Breeders. We are representing more than 40 companies and associations involved in pigs, poultry, ruminants, pigs, poultry, ruminants, aquaculture, and insect breeding. I'm sorry, I have to go back to the presentation. Okay, I'm going to in another place. So FABRE-TP is a sister association of EFFAB in which we represent animal genetic scientists across Europe, and all the EFFAB members are members of FABRE-TP. FABRE-TP intends to build bridges between the private sector and the public research in Europe in animal genetics and reproduction. So we are involved in policy at European level, not only, but mostly at European level. We are involved in many topics currently related to animal welfare, to sustainability of food systems, and also, of course, around genome editing and the legal status of new breeding techniques. So we are, as I said, we are connecting industry and knowledge institutes, but we are also supporting and promoting responsible and balanced breeding through a code of good practice, which is called Code EFFAB, which should be after in the slides. So EFFAB is also currently involved in European funded projects. We are a knowledge provider in these projects. Most of them are related to genetics, to animal genetics, but not all of them. I mean, roadmap, for example, is about antimicrobial resistance, but of course related to animal production in Europe. Within Aquafang and GenSwitch, there is also BovRec, in which we are not involved, but the 3 of them are involved in the FAANG initiative, which Emily will explain later today. Let me remind you the house rules for today. If you have any question to the panelists, please ask the question in the QA box starting now if you have questions already. But if you have any technical question to the team, or you're experiencing technical issues, please use the chat box, or you can also mail our colleague Norali. Please note that the webinar is going to be recorded, and we will share the video and inputs given today in our social media. And of course, in a couple of days or week or next week, we will share with you a summary of the discussions, the presentation and discussions that we are going to have today. So I was telling you that we are promoting responsible and balanced breeding. Emily will talk a bit about it later too, but just to show you that this is called FABRE, this code of good practice, in which we try to show, to make more transparent the animal breeding sector in Europe, and including these 6 pillars, which are the main traits that are in modern animal breeding programs nowadays. So the welcome is done. I would like now to introduce you our first speaker, which is Mr. Christensen, the member of the European Parliament. He is also a farmer in Denmark. He is involved in the work of the Committee, the Agriculture Committee at the European Parliament, and he has been quite active member at the ANIT Committee. The ANIT Committee is a committee of inquiry. This committee has been working for 18 months at the European Parliament to take a look at the implementation of the European animal welfare legislation during transport of the animals. So Mr. Christensen has been also been involved in the debates at the European Parliament on the about the European strategy to reduce methane emissions. And so let me give you the floor, Mr. Christensen. And yeah, we are happy, we are delighted to see you today with us. Thank you very much.
Thank you for the introduction, and thank you. I can have a short speech here from the Copenhagen airport. Yes, as you said, I'm farmer. I'm dairy farmer in Denmark. I have been dairy farming 35 years, and we have 650 dairy cows on my farm, and has been involved in politics in many years in agri-politics, and now the first time from this mandate in European Parliament. So I have a lot of experience from the farmer views, and I'm using my farmer views in my politics, and I try also to think about how the politics can work on my farm. And of course, it's for all of European farmers, as I'm thinking. But in my mind, I try to use it that way. And as you know, it's very good to sometimes bring the real life into the politics in the political discussions. It still works. So therefore, of course, it's my focus is in agriculture, and but it's not only food production. My ambition is also to guarantee a high quality of food supplying in the future, and at the same time to reduce emission from the agri, and also we have a strengthened biodiversity. Maybe it sounds a bit too ambitious, but I think I believe it will be possible. As you told, I was a shadow of Farm to Fork strategy, and also was a rapporteur and chief for the European Parliament's Agri Committee on the methane strategy. So I have worked with the green future in agri. And one of our politics highlights in the methane was we shall reduce the emission, not the production. And to reach our common climate and biodiversity goals, we need to use all the tools, such as a simple circular bioeconomy and also CRISPR. It will be we shall use CRISPR in the future, and we have a good discussion last week in the Agri Committee about CRISPR, and I hope we can get a result of that in the next very, very soon future. And also, of course, we must discuss and use GMO also. I think we shall use all the new technology for the most effective, and also, but I also know it's maybe for some of our colleagues in the parliament it's not politically correct, but we must take that on board. And we have just discussed the vaccines of COVID, where we are using GMO a lot. So I don't understand why we not can use it on the breeding also. And we have to look at animal breeding also. If we show can we have cows in the future who produce 10% less methane, example. I know there is some cows that can do that, And I'm quite sure we will find that. And I am also in my 35 years of dairy farmers, we have breeding a lot of new roads, and now we are using genomic selection also, and it can be a game changer for us to be sustainable in the future. So I'm very excited to follow the development in animal breeding, and because I believe that can be a very important tools to a greener agri. And also that you in your introduction explained that I'm also being a member of Animal Welfare Transport Committee, and I hope we will get a good solution for that in the plenary in January. But it has been a very, very hard work, and there is also a lot of feelings for some some of my colleagues in the parliament. But I hope we will find a good solution for that in the future, and also for the farmers, because we need to transport animals for new breeding. We need to transport animals for slaughtering, and of course, it shall be on the good way. Thank you very much, and have a good webinar. Thanks.
Thank you very much, Mr. Christensen. We know that you have to leave us because
I have 3-4 minutes if you want.
Okay, so perhaps we can ask you the question we have in the chat for you. Please use the QA questions box, but well, we are going to take it from Karel de Graaf from Wageningen University, if I'm not mistaken. He's asking, you have mentioned CRISPR tools during your speech. He's asking if you have said we will use CRISPR, is that a Danish dairy farmer is aiming to do, or is it just talking about European policy need to be improved?
CRISPR will be one of the tools we can use in the future, and what I said in my speech, I hope we will get a solution very, very soon, because a lot of companies, a lot of universities in Europe are waiting for our solutions, and they are just begin to research in that technology in US and in China. And for me, it's very important we make a solution in Europe for this new technology.
Okay, so there is an additional question, if you're still here with us. It's again about new breeding techniques, about genome editing, and this can be used in a responsible way by breeding associations and company. But the question is about the public perception. So the majority of the public still see this as being a Frankenstein science. So what do we need? What do you think we need to do to improve knowledge and understanding of this technology?
I think we shall try to tell it's a natural way, but it's a faster way than the nature is using. I'm explaining that way. We speed the time 10 times up when we're using CRISPR instead of natural way. So we are getting faster. So it's a faster way than the nature is using.
Okay, thank you very much for coming. We know you have to take a flight, to come to Brussels from Denmark. So thank you very much for coming today to be with us, and have a nice flight to Brussels. And thank you again. Thank you very much. Yeah, bye-bye. So Emily, we can see you. You are spotlighted now, so you are going to share your screen, but let me introduce just a few words about you. So Emily, again, as I said, she's a scientist from Roslin Institute in Scotland. So her main research interests focus on transcriptomics, and genomics in livestock, in both production animals and indigenous African breeds, if I'm not mistaken. She's particularly interested in functional annotation. That's the presentation from today. And she is involved in the FAANG initiative, which is involved in the Gen Switch, FABRE-TP, and Aquafang projects. She's one of the authors of the stimulating article published last year, From FAANG to Fork, FAANG to Fork. And this is what she's going to talk about today. So I give you the floor, Emily, now. Thank you very much.
Thank you, Anna, for the introduction. I'll just share my screen.
Yeah, I have stopped sharing mine, so you should be able to share yours.
Thank you. Yeah,
perfect.
Okay. Okay, great. Yeah, thank you, Anna, for the nice introduction. So today I'm going to be presenting the FAANG to Fork strategy, which is a research strategy or vision of a core group of researchers in the global FAANG consortium, which is Functional Annotation of Animal Genomes Consortium. And this is a vision for the next 10 years of research, focusing on how more knowledge of the genetic code of farmed animals will benefit animal breeding. So first, I would like to introduce what animal breeding is and why it's important. And I've been kindly lent a slide produced by EFFAB for code FABRE for this purpose. And code FABRE, as Anna mentioned earlier, is a code for good practice in animal breeding. So a breeding program for many farmed animal species, such as chickens, cattle, pigs, and sheep, involves making breeding decisions based on a balanced and responsible combination of traits or characteristics. So animals can be bred for different traits or characteristics related to health, welfare, productivity, and sustainability. This is a complex and dynamic issue, and achieving the right balance of traits in a responsible way can be informed by recent research. And a better understanding of the genetic code of farmed animals is exactly the right kind of research for this purpose. And it can help farmers and animal breeders breed healthier and more productive animals. For example, information about the genetic code of farmed animals can inform genomic selection, as we heard the previous speaker mention, and genomic selection can improve important and very complex traits such as resistance to disease and environmental impact. So breeding programs consist of many animals with different genetics and characteristics. Currently, genomic selection is used by some farmers and animal breeders to make breeding decisions based on an animal's genetic code and also its characteristics. And one of the key things to do is to associate the genetic code with the animal's characteristics. And this information is then fed into a prediction equation that allows farmers to breed the best animals for their particular situation. So the more information we have about the genetic code, we can make better and more accurate predictions. And this better prediction accuracy will lead to healthier and improved animals. And these can be, for example, for the traits I mentioned previously, such as resistance to disease or environmental impact. So chief among the improvements required in animal breeding in the coming decade is to more accurately use an animal's characteristics or its genetic code, an animal's genetics or its genotype to predict its characteristics or phenotype. And the FAANG to Fork research strategy lays out a framework for research linking genotype to phenotype in farmed animals for the coming decade. And it has three main components. The first one is to generate more knowledge of the genetic code of farmed animals with information for many different breeds and populations to improve genomic selection accuracy for complex traits such as disease resistance. The second component is to build maps of gene expression. So this means how genes are switched on and off, and gene regulation. And this is the parts of the genetic code that operate as the switch to switch genes on and off. And this is important because it tells us which parts of the genome of the genetic code are controlling which characteristics in farmed animals. And then the third component is to provide information about fundamental biology, so linking cell, tissue, and whole animal scale knowledge, so that we can understand where and when during development, for example, elements of the genetic code are having an effect on an animal's characteristics. So together, these components will inform genomic selection, also inform genome-enabled management, where we could envisage a situation in the future where very high-value animals in breeding programs have their genome sequenced, and then management decisions are made for those animals based on their information about their genetic code. They would also feed into information about fundamental biology, which I've just mentioned. And then this kind of information can also be used for new breeding technologies such as genome editing. And genome editing is one key route to application of more information about the genetic code of farmed animals. And together, all of these components will lead to healthier and improved farmed animals in coming decades. So this is the farm to fork research strategy in more detail in this figure. And in this diagram, you can see 6 of the research priorities that we identified for research for the next decade in this space. So research particularly looking at the genetic code of farmed animals in different ways. So many of these research priorities are already components of an animal breeder's toolbox. So generating large phenotype collections, having information about lots of animals in a population. But then there are other priorities that can help us to improve understanding of fundamental biology, such as looking at gene expression and gene regulation across tissues, and also using genome editing technologies to make small changes for example, in cell culture systems, where you can look at how small changes can have a fundamental effect on the characteristics of those cells or animals themselves. So several other of the priorities are also important to discover and utilize biodiversity. So for example, having information for the genetic code of multiple breeds within a species or multiple populations of animals. And that can help us conserve and utilize biodiversity. And another way that we can do that is also through biobanking. And biobanking is also a resource for research and conservation, where we biobank tissues and cell types from different breeds and populations of animals. So the outcomes of these research priorities and more generally the outcomes and benefits for animal breeding of having more information about the genetic code of farmed animals are, as I've mentioned on previous slides, enhanced genomic prediction accuracy, particularly for smaller breeds or breeds that we don't have very much information about the genetic code for already. With more linked genotype and phenotype information, farmers and breeders can make better decisions about managing their herds and individual animals within their herds and populations. So by understanding more about the genetic code of different breeds and populations of animals, we'll be able to better conserve biodiversity and utilize and preserve biodiversity as well at regional and global scales. Together, all this information will ultimately lead to improved farm animals for health, welfare, and production efficiency traits, particularly those that are complex, such as disease resistance or environmental impact. And this could be through the application of new breeding technologies or simply through making informed breeding decisions. These outcomes will help to meet the goals of the sustainable food production in the farm to fork strategy, which is part of the EU Green Deal, and lead to healthier and improved animals. But public investment is needed to expand genotype to phenotype research in farmed animals and train new scientists in this space, particularly in terms of data and data management. So now I've talked to you about the potential outcomes, why information about the genetic code of farmed animals is important. I'm just going to talk about, at the end, a couple of slides describing public investment so far in this space. So there's been significant investment already from the Horizon 2020 research framework. So EFFAB is a coordinated effort to create a suite of free resources that link genotype to phenotype to improve animal production and welfare. So the EFFAB community is currently supported by 3 key projects: AquaFANG, which focuses on aquaculture species, BovReg, which focuses on cattle, and GeneSwitch, which focuses on chicken and pigs. And these projects lay the foundation for the FANG to fork research strategy and for FANG research for the next decade. And this is in line with a new era of farmed animal production based on biological efficiency, precision breeding, disease resistance, reduced environmental impact, and feeding a growing population. So EFFAB, as I mentioned, is a synergy of the 3 Horizon 2020 projects, AquaFANG, BovReg, and GeneSwitch. By establishing EFFAB, they've formed a closer relationship to coordinate their objectives within Europe in close association with the global FANG initiative. The initiative brings together a wide range of expertise in farmed animal biology and breeding, also genomics, bioinformatics, modeling, and open data access, as well as multiple platforms for dissemination and outreach, with a common goal to discover links between genotype to phenotype in farmed animals. This year, 2 new projects, Geronimo and Rumogen, which Anna mentioned at the beginning of this session, started, and like GeneSwitch and BovReg, they focus on genomics in cattle, poultry, and pigs. And they would be a very valuable addition to EFFAB should they decide to join. So I just described where EFFAB is at the moment, and thinking to the future, we're now building on a foundation laid by these Horizon 2020 projects to develop a formalized meta-infrastructure for genotype to phenotype research in farmed animals in Europe. And this would also be titled EFFAB. So this meta-infrastructure would be developed according to user-defined priorities that are outlined by stakeholders, including animal breeders, exploit synergies with existing infrastructures, including PigWeb and SmartCow, and would streamline how we best use our capabilities in genotype to phenotype research in farmed animals across Europe, and to achieve the research priorities that we laid out in the FANG to fork research strategy. So I'm just going to finish there and point your attention to the FANG to fork publication. I'd like to thank all of my co-authors on that publication, the Horizon 2020 coordinators and co-coordinators, and particularly Elisabetta Gufra, who's also listening to the seminar, I believe. And thank you very much to Anna and the EFFAB team for their help with putting together this presentation. And thank you all for listening.
The next speaker, I have just one question from the attendance about what is FANG. So I'm going to start sharing the screen again, but if you can answer just to this simple for you question, it will be great.
So FANG is the Functional Annotation of Animal Genomes Consortium, which is a global group of scientists with the shared goal of initially improving the reference genomes of the farmed animal species and animal species generally, but is now moving to a new phase where we're looking at genome and phenome information for populations of animals. But there's a European sort of EFFAB, and there's a US and Australia. So it really is a global initiative for genomics research in farmed animals.
Great, thank you very much.
Thanks.
So our next speaker is Javier Ojeda, the Secretary General of FEAP. Can you unmute yourself?
I'm here, yes.
So, yes, hello, Javier, hola. So Javier Ojeda is the Secretary General of the European Federation of Aquaculture Producers. He is also the chair of the Aquaculture Advisory Council. At the European level is a stakeholders group in which we discuss and we give our impressions, our perspective, because EFFAB is also a member to the European Commission and topics related, of course, to aquaculture in a broad, in a large spectrum of aquaculture. Javier is also representing the aquaculture sector in Spain through APROMAR. And he is here with us today to share his views about how breeding can help aquaculture, can help, or what's the challenge for breeding to fit with aquaculture producers' needs. Thank you very much, Javier, and the floor is yours. And stop sharing the screen so you can share yours.
All right, thank you very much for inviting me to be here with you and share the views of aquaculture on this field. My presentation is not as technical as the previous one, but I'll provide our general views on where we stand and where we think aquaculture can take us for the EU sustainable food systems and for achieving the United Nations Sustainable Development Goals in Europe and around the world, of course. Okay, so FEAP, we are, as Anna has mentioned, the Federation of European Aquaculture Producers, and we are composed of 20 national associations from 23 countries in Europe, not only in the EU, but other countries outside the EU. Aquaculture, one of the characteristics of aquaculture is that it is very, very diverse in the number of species and also on the types of systems used to produce these species. So in FEAP, we represent finfish, and in our species there is salmon, Atlantic salmon, rainbow trout, sea bream, sea bass, turbot, sole, meager, sturgeon, Arctic char, carp, eels, and several other species that all together provide to society 2.5 million tons of food that is highly nutritious, of course, safe, and also delicious, but at the same time, environmentally sustainable and other aspects of sustainability, of course. So aquaculture has grown very, very fast in the world in the last 4 decades, not so much in the European Union, but in the world, it has been the fastest growing food sector in considering all of them. And today, farmed aquatic products bring for human consumption more quantity of food than captured fisheries, for example. And at the same time, it's a very, very internationally traded food product. Considering the growth, today's huge population on Earth, but also its expected growth until 2050 and probably until 2100, feeding represents major challenges to humanity. And not only considering the challenges that we had until now, but also adding now the climate changes that are occurring. Given especially the nutritional value of aquatic products, aquaculture is considered to continue being an increasing important need for supplying food from the seas, from the lakes, and from the rivers to provide healthy diets and provide, on the other hand, livelihoods for millions of people, and alleviating at the same time the pressure on wild stocks, on the natural environment, and this is as FAO states. So considering the Sustainable Development Goals of the Agenda 2030 of the United Nations, aquaculture can contribute to many of them, but let me bring it to your attention, especially two. First one, number 2, is about hunger, and development goal 14, about life below the water. It is true that Europe does not lead world aquaculture production in volume, or say in tons, but it does do it in technology, in efficiency, and environmental sustainability, and other aspects of sustainability, like fish welfare, for example. Now, aquaculture genetic resources. While aquatic genetic resources are today a very valuable reserve of biodiversity, it is highly unexploited until today. Today in the world, there are around 600 species farmed, but that's very little compared to the 50,000 finfish species that exist in the world. Farmed aquatic species besides finfish include mollusks, crustaceans, and then plants, vascular and non-vascular, and at the same time many microorganisms. But today I'm mainly going to focus my presentation on finfish. Many of these organisms, most of the farmed species in aquaculture, their production cycle still depends today on exploiting their wild counterparts, meaning that wild relatives of many aquatic genetic resources are still today collected from the natural environment to be bred and to be raised under farm conditions, including in the European Union. So even today, the aquaculture sector remains very closely linked to wild aquatic genetic resources and their habitats. There are a great range of genetic technologies and tools, like the ones that have been mentioned earlier today, that can be applied for aquatic species, just like for terrestrial livestock. In aquaculture, the traditional approaches of selection, hybridization, and crossbreeding can be applied, but there are also means for manipulating ploidy and sex. Notably, I should mention that the first transgenic animals produced for commercial food for production were fish. As FEAP, we do not endorse this food, but it's a fact, and I need to mention it. Then another important point here is that farmed aquatic species, or aquatic species in general, I would say, have significantly larger reproduction rates than terrestrial livestock, meaning that one fish lays every year millions of eggs and have a relatively high survival rate. Some technologies can be used for immediate short-term gain, where others are offer more longer-term gain. But what we can always get is genetic improvements accumulating over each generation. And although the new gene editing techniques are emerging, can be applied for the farmed aquaculture species, they have not still today applied widely in commercial aquaculture. Well, not even widely, and not very little in aquaculture. We're still working on them. The basic requirement for the application of all genetic technologies is the ability to reproduce the species under controlled conditions. And as I said before, even today, important species in aquaculture like eels or bluefin tuna or mussels are not carried out in full cycle under aquaculture conditions. Genetic improvements in traits by selective breeding can produce genetic gains of about 10% per generation. Aquaculture geneticists have stated that if all farmed aquatic species were in traditional selective breeding programs, improvements in aquaculture production efficiency could produce a doubling of aquaculture production by 2050. And this is important because that is precisely the growth that FAO expects or plans aquaculture should reach to meet the projected increase in demand for fish and fish products, with a low proportional requirement of additional land use, water use, feed, or other inputs. Building knowledge and facilitating access to that knowledge is considered essential to raise awareness and address the main challenges, the main needs and advantages for aquaculture breeding and genetics. And here we have a list of challenges for aquaculture that are accelerating the genetic improvement of key aquaculture species. The second, developing and promoting effective access and benefit sharing measures. A third one, to address the threats to the natural reservoirs or diversity of wild relatives of farm species. A fourth one, improving or say implementing well-designed and integrated conservation programs. As I said, aquaculture is still today very linked to the natural environment and the species that live in the natural ecosystems. And the last one, supporting the development of adequate governance systems. And my last slide, I have made a list of the genetic technologies that can be applied in aquaculture. I mean, the genetic technologies that can be applied. The objectives would be to increase production, to control reproduction, to improve marketability, to be able to have more accurate and effective traceability along the supply chain, for better disease and parasite resistance, and for more efficient utilization of the natural resources as inputs into aquaculture. I believe these are basically the same for other livestock production, but as this is the one in which I am, these are the one that count for us. So as we like to say, aquaculture is a solution for the future. Important part of the solution, even in the European Union, to reach our sustainable food systems. Okay, Anna, so this is up to my presentation, and I'll be here available for questions that might come up. Thank you very much.
Yeah, thank you very much, Javier. So now we have, you should stop sharing your screen, Javier.
Yeah, I'll do that.
Don't worry, we are good until now on time. We have a question for all of you before going to the panel discussion. I will introduce our last, the person who is going to join us for the panel discussion. But first, we are going to you have a in the banner you have a pool. And we have a question for you that I cannot see anymore. Yes, exactly. So yeah, I have seen that some of you have started already to answer. So what is the main challenge for the new genomic research and next level animal breeding to be used in Europe or European Union in a broader way by the breeding companies and farmers? So there is only one choice that you can click on. So we are going to wait some seconds or 1 minute to see who is responding. And already societal perception is first choice of more than 60 people voting at this moment. Yeah, it is not changing a lot, almost, yeah, not almost, now 80 response. Yeah, that's societal perception is the most important one, more than 40% of the response. Just a second, we are going to wait a bit more because people are still responding. So thank you very much. Okay, we have 95%, 95 response, sorry. And there is talking, yeah, and this is almost the same. So 40, more than 40%, 42% thinks that is societal perception. And after around 15% for affordability, practicability, and regulatory framework. So yeah, thank you very much. We're going to stop sharing this, and we are going to introduce our our new panelists. So now we have the panel discussion. We are going to address your questions. So you can continue to put your questions in the QA box, please. And so Emily has joined us again for the panel discussion. Javier is staying with us, and we have also Peter Stevenson from Compassion in World Farming, who should join us. Peter, can you unmute yourself?
Yes, hello, unmuted.
Perfect. Thanks for coming today to have this discussion with us. I'm going also to introduce my colleague, Çağla Kaya. She's also, her camera is also open. So we have now the discussion between you, the three of you. Mark is also somewhere. So Mark, I know you are not participated in the panel discussion, but if you want to give some perspective from the breeding sector, don't hesitate to do it. So yeah, Çağla, the floor is yours.
Thank you, Anna.
Here to help, of course.
Yes.
If you need.
First of all, I would like to thank Emily and Javier for their nice presentations, very easy to understand and follow. But I would like to give the floor to Peter first, and I would like to learn the standpoint of the Compassion in World Farming on these new genomic research and its application in the commercial farm populations.
Hello. Yes, my opinions, and I've just spent the last 3 years on the working group for the Nuffield Council report published last week on gene editing. My concerns are to do with the fact that over the last many years, 30 or 40 years, just traditional selective breeding, genetic selection, have caused immense animal health and welfare problems amongst broilers, hens, dairy cows, pigs. And therefore if I think about farm animal welfare, for most of the public, the main thing they're going to think about are cages, crates, extreme overcrowding, and very rightly so. But I think selective breeding has caused just as many health and welfare problems when you think of the level of bone fractures in laying hens, the many problems in the pig sector from large litters. And so if I then think about new breeding technologies, genetic selection, gene editing or whatever else may be coming along, I'm extremely wary. I feel that they're in the main are going to be used to drive increased productivity, and that will push animals to even greater health and welfare problems. They'll also be used to drive disease resistance. And in some cases, that can be very good if the diseases have nothing to do with the way in which the animals are farmed. But a lot of animal diseases are coming precisely from the way in which the animals are farmed, from keeping them in very crowded, stressful conditions. And creating disease resistance in those cases could lead to animals being kept in even worse, more stressful conditions than at present. And I was a little bit disturbed that none of the speakers so far have addressed the animal health and welfare problems and thought about, okay, where should our farming be going? How should we be meeting the challenge of improved animal welfare that the European Commission clearly wants to see? Thank you.
Thank you. So I will now like to give the floor to Emily about this, because I know that there are also some tools that could be used for management of farmed animals for FAANG research. What kind of new opportunities could they provide for farmers, especially in addressing the topics that Peter highlighted?
Yeah, so it's, I suppose there are many ways to answer this question. So having more knowledge of the genomes of animals provides a tool in itself. So we can understand more about the regions of the genome that are important for welfare traits. We can look at more about how heritable welfare traits are, which I think is important. So how they're transferred through generations, and that we can breed through breeding decisions based on genomic information for better welfare. I see Peter's point about the way we are farming animals and the link to disease based on the farming system. But I still think having an improved amount of information about the genome can help really tackle disease. So if you look at the example, the Powers pig example, where the gene CD163 was annotated and then led to editing here at Roslin, actually, to produce pigs that are resistant to Powers, which will have significant benefits to pig production eventually. But obviously, if you're managing disease better, you're feeding animals less, there's less input for the farmer and better welfare. But I think the question of how we manage farming systems isn't necessarily a question unfortunately, for FAANG, a bigger question. But we can provide tools to help.
Yeah, thank you, Emily. So Javier, I want to return back to the aquaculture. And how do you think these research contribute to the objectives of sustainable and competitive European aquaculture guidelines, also taking into consideration the concerns of the NGOs that one is represented by Peter here?
Sorry, can you repeat again the question?
Yeah.
Because I lost some.
How do you think the research could contribute to the objectives of the sustainable and competitive European aquaculture guidelines?
All right, thank you. Good point. Okay, research is absolutely essential. Scientific knowledge is the foundation on which we do all our work. So we know there are many gaps still in knowledge on aquaculture. So from our federation, we encourage all the research efforts in fields like fish welfare, for example. We work quite intensively with compassionate world farming in particular, because we know how important it is. And we are aware of not knowing enough to do things as well as we would like. So research is basically, answering directly your question, absolutely necessary to reach the objectives of the strategic guidelines. But I would like to go to the previous question from Peter to Emily, well, that you addressed to Emily. And okay, aquaculture is a little bit different to other livestock because we've been very little time producing our animals when compared to other livestock. But I do come from, as a general comment, not only on fish, I do come from the countryside, and okay, we can see livestock production today as a certain way, but I can tell you that at least in Spain, the way in which animals were treated 50 years back was awful. I mean, brutality on animals there was absolutely no sensitivity towards animals. Today it's a completely different situation, in which, of course, we can always look at specific aspects of a huge industry today, but I don't think that in the past things were any better. In fish, what we are certainly trying to do is to build upon we know right now, to offer our animals that are under our responsibility the best welfare conditions. But of course, at the same time, we have to take into the consideration that we will have to improve other aspects of production at the same time. Thank you.
Thank you, Javier. Maybe I have very few minutes, so I can ask another question to Peter by asking, well, do you think breeding supported by new management tools could be a solution in addressing some of these challenges that your community is concerned about?
I think breeding can only be of help in addressing the kind of challenges I sketched in if there's really a quite fundamental rethink about where we have got to. Although breeders will very often say that they're now including health and welfare amongst their objectives, and I'm sure they are, it's not going far enough. We need to get to the point, for example, where we are breeding for much slower growing broilers, just to take one example. One of the comments in the chat has said, well, the problems of fast growing broilers have been much reduced, but that's not what the science has been showing. There were 2 studies, one of them, I think, from Edinburgh last year, showing that the kind of problems of fast growing broilers remain as severe as ever they have been. We need to get to the point where litter sizes in pigs start going down, because it's just simply not possible to keep sows or piglets to high welfare standards with very high litter sizes. So, you know, if you look at the farm to fork strategy and the recent inception impact assessment the Commission produced on welfare, really ambitious things for welfare. They're talking about more animals being kept outside, lower stocking densities, you know, more space. We need quite a fundamental rethink. So breeding can play its part, but it's got to move away from this incessant trying to push animals further. I went onto the website of one of the world's and Europe's biggest pig breeding companies recently, and you know what they're offering customers is, we can ensure that you have even more piglets per sow per lifetime. We need to pull away from that pushing of animals so hard. So I think breeding can play a part, but it would need a really quite big rethink about the role of breeding in modern farming. Thanks.
Thank you. Well, Mark, maybe you can just get into the discussion by saying, how do you think that this research and research could be uptaken by the industry? Like, what are the main challenges or opportunities that you see for the breeding sector, while also addressing these kind of societal concerns?
Thank you, Çağla. Yes, I think as Emily has highlighted, one of the main areas that's of great significance is disease tolerance or resistance. You know, we know so little at the moment about the genetics and the underlying disease susceptibilities and resistance. And with the help of FAANG and looking at the phenotypes and understanding the function on how these genes interact, will provide us with a much better understanding of how we can use these in breeding programs. Yeah, I think that's my perspective. Thank you, Çağla.
Thank you very much to all of you. Now I give the floor back to Anna for the audience, the questions coming from the audience.
Yes, thank you very much, Çağla. I was just texting you about this because I think you were making a very good job. So so you could continue to the questions coming from the QA box. So stay with me, please. We have a number of, so we're still with our, no, go back, Isabella. The wrap-up is after. Yes, we can stop still with the, so we have quite a number of questions. There is a lot of questions for Emily, but not only. So there is a question from Roland. This is already, he has a conversation with Harvard about the feed of salmon. So if there is something that we can bring here, we will do it later, but I think we can go to the questions from the audience. It has been mentioned several times now. It is not the first question, but it's one of the questions around. It is to Emily. Do you have, and we can also ask to Javier and Peter, of course, but first for the scientists from a researcher's perspective, do you have an example of disease resistance traits in mind which could be improved? And could you explain how FAANG will help in this way? Whereas we don't have the knowledge of the underlying genetic control of resistance to diseases, which are mostly polygenic. You have mentioned PRS before, but have you got any other examples?
Yeah, so there are a few examples. So the PARS virus one that we just mentioned, also IPN in Atlantic salmon is another example where the loci has been fairly well unraveled with genomic information. Another one I can think of is from collaborators in Bovreg. So Carole Charlier and others in Belgium identified a mutation related to mastitis resistance that's a regulatory mutation, an actual copy number variant in dairy cattle that explains to a large proportion of resistance to mastitis. So we're just, these kind of studies do take a long time, and we now, and you sort of need to, it's difficult to get the perspective of how much genomic information is required to get down to this level, and how much effort goes into producing these studies and getting to a loci or a copy number variant that's actually controlling resistance. But there are some examples and many more coming, I think.
Yeah, Javier, we move to aquaculture. Something you would like to mention related to disease resistance or to improve animal health? You were mentioning already during your presentation what is some of specific challenge to aquaculture diseases. Perhaps you can mention.
Okay. Yes.
And just a second, make a follow up of this question. Is that so we have this discussion about disease resistance, but there is a comment saying talking about there will be more strategic to improve immunocompetence, robustness, resilience. That's a part of course. There is no one solution, I guess, but just you can perhaps make a follow up also with this comment, Javier. Sorry.
Yeah, thank you. Well, on disease resistance, I would say that being able to have more robust animals in general, make them more fit for farm conditions is it would be the first point. Okay, in fish, we are so behind on the domestication of the species that when I listen to all the work that is being done on other species, I really feel very humble because we're sort of very much behind. And actually, one of the points of my presentation was to explain that we are very close to natural species right now, to wild species right now. And the main challenge, or one of the main challenges of the work that we do on fish breeding and genetics is not so much on the welfare of the animals, and I do appreciate Peter's presence here. But it's more from an environmental point of view. We are very concerned, and there are societal concerns on what happens to the fish, to farmed fish when they escape, or they can be escape for different reasons from the farm. So that is very high on our agenda, and we receive many comments from other stakeholders on this and how we can solve this issue. That for terrestrial animals, it's completely behind centuries ago. So that's where we are looking into mainly now, and the concerns that society has on this. And of course, I've read some comments on fish meal, fish oil on the chat. Well, I wouldn't say that. I mean, fish breeding is not the first, or the genetics is not the main tool for the design of the diets of the fish right now. We have other scientific tools and disciplines to deal with this. So that would be my comment right now, and thank you.
Okay, so Peter, I will be happy also to hear your views about, because you were saying that we have these tools have the potential to help, but is they used in a responsible way? What do you think about tackling disease resistance?
As I said, yes, I mean, clearly tackling disease resistance can be very helpful, though, as I also said, one's got to be careful. One thing that came out very strongly in the Nuffield Council report was the danger that disease-resistant animals could be kept in even more crowded, intensive conditions than at present. I like the comment from somebody a couple of minutes ago about perhaps we should rely more on developing immunocompetence. There was a body called the Lancet Infectious Diseases Commission a few years ago developed the term, they said we need to develop health-oriented systems for rearing animals. They were saying rather than being so reliant on routine preventive use of antimicrobials. And I think if you look at the science, health-oriented systems would be ones in which there's much less stress for animals, in which they're able to engage in their natural behaviors. We avoid overcrowding or very large group size. For pigs, it would certainly mean an end to early weaning. This is where so many of the problems and so many of the use of antimicrobials comes. And given the EU ban on routine use of antimicrobials comes into force next month, we need to very quickly think about how our farms can respect that ban. And I think we really do need to move towards health-oriented animals. So yes, I accept that breeding has a part to play, but we shouldn't just rely on that. We should be thinking about how to develop farming systems in which good health is inherent in the farming methods. Thank you.
Okay, thank you very much, three of you. There is a there's a lot of questions in the chat.
Maybe we can.
Yeah.
Maybe there is this one about this changing environments.
Exactly.
So more
Go
adapted
ahead,
animals. So there were a couple of questions asking at the same time, like, is it like efficiency versus like adaptation or changing environments? How do you think, or which kind of traits do you foresee for the changing environments? How can we use these technologies to increase the adaptation of animals to changing environments? Because this also has a role in animal welfare, I think, and health, of course. It is an open question for the whole panel.
Yes.
Mark, maybe you can share your views as a representative of the sector, of the breeding sector. How do you tackle with this coming or future challenge?
Yes, I'm unmuted. Thank you, Çağla. Yes, I think the looking into the future with environmental challenges and change in climate will probably have less of an impact on poultry, but animals that are outdoor reared, cattle, sheep, pigs, so in some cases, I think it may well impact more in the short term. Is that the sort of question you were thinking of, Çağla?
Well, yes, and maybe to more extent, because there is like there are the questions which consider that, okay, we are going to select some traits and which are important for the commercial production, of course. When you put it in a balance, which do you think will be more important in the coming years?
Well, I think you need to bear in mind that current breeding goals are much more widespread than they were in the past. I mean, the welfare and disease traits and environmental impact are major goals that we have as in looking at breeding values and genomic selection. So I think it'll be put into that algorithm and that analysis that we do for future breeding goals.
Thank you. I think it is important for the audience to know that the breeding companies are already ahead of thinking about this and trying to make a balanced and responsible breeding strategy. Any other comments from other panelists, or maybe Anna? Oh, Peter.
I just want to look at another environmental aspect, because again, what I think what I'm really wanting to promote is this idea that when we think about breeding, we need to think about what kind of farming system do we want in Europe and globally in the years to come. And some of the biggest environmental problems that we face globally are coming from our very industrial livestock production systems, and they're coming primarily from this huge use of cereals and soy as feed. All these very intensive systems are very dependent on using soy, and yet we've now got a proposed deforestation regulation in Europe, which is really trying to end the import of soy. The Commission itself states that almost 2/3 of EU cereals, wheat, barley, oats, maize, are used as animal feed. Study after study shows animals convert this very inefficiently into meat and milk. We need to pull away from the, and yes, the point I should make is that it is this huge need for cereals as feed that has fueled very intensive crop production with use of agrochemicals and monocultures. And that has led to soil degradation, biodiversity loss, overuse and pollution of water. So many of the goals of farm to fork can only really be reached if we rethink the livestock sector. I really want to emphasize, I'm not against livestock. We're not a vegetarian or vegan organization, but really animals only are making an efficient contribution to food security when they're converting materials we can't consume, like grass, food waste, byproducts, into food we can eat. So we need to think about breeding in the context of thinking about our food and farming systems and its future as a whole. Thanks.
Thank you. Anna, maybe you have questions.
Yeah, and there's also a question. Peter was talking about the future of farming. There is a question for Javier mainly, but could be answered also by the rest of the speakers. You were mentioning, Javier, that we could expect a difference in public acceptance for genome editing tools, and you were mentioning that one of the main points for aquaculture is to manage the escapes from farms when it happens, and that there's one of the challenge. So, how do you see these techniques to tackle this problem of escapes, to avoid mixing farm species in aquaculture with wild ones?
Yeah, there are two approaches to this. First, to prevent escapees, right, through better engineering, better structures, better operating, better training. And then if some fish escape, we have to do our best that those do not affect or to minimize their impact on the natural stocks. The direct way to that is to farm sterile animals, or animals that are not capable of reproducing, but also that are not capable of surviving in the wild. And in general, they are not capable of surviving in the wild, but that has to be reduced as much as possible to prevent the genetic contamination with their wild counterparts. So that's how we approach this. And just going back to what Peter mentioned, I think that all of us here are rethinking the future of livestock in the EU, in the world. I mean, that's why we all work in this. We are all very, very concerned, and I think that the concept of sustainable food systems is very powerful, and all of us have this in mind. Of course, we have different approaches, different views, but we know that we have a huge challenge in front of us. And luckily now we have science to work with, science to progress. But just to say that we don't want to stay where we are. I don't think nobody wants, not because we're in a bad position, but because of the challenges ahead and climate change and the increase of demand of food in the world. Thank you, Anna.
Thank you, Javier. I was going to add to that, actually. Yes, I think one of the points that have been raised is the public perception issue, which, that's twofold. The public perception of gene editing is actually generally, for gene editing itself, is not that bad. I mean, if you look at human medicine, where we've used the understanding of underlying genetics to actually go as far as, curing specific diseases. Obviously, this is done in somatic cells rather than the germline. But on the other hand, it is the public that we need to get on board to realize that these are the sustainability goals that the breeders have, are something that they must be aware of. The bottom line is, if people want cheap food, that will be supplied. Whereas we need to get on board the public that things may cost a little bit more in the future for embracing the sustainability goals. So thank you.
Okay, we have a last question. I have to go from the QA to the chat. I'm sorry. We have a question, and this is part of the EFFAB, I guess. But so it's a question more for Emily. It's on the in Davos at the AAP meeting this summer. We spoke about the need of breeding animals, more regional, different, that fit into circular farming. So for regional, so can genome technology help there in any way? You were mentioning biodiversity. Or what technology do we need for that? Javier was also mentioning something, so you can, we would like to jump after.
Sure. So I can start. So really, from a FAANG perspective, the next stages of FAANG will include generating genomes for more breeds within a species, and that can include local breeds, indigenous breeds, breeds that would be included in these circular farming systems that are region specific. Because at the moment, or previously, we've kept very highly annotated genomes to usually to very specific managed production animals, and understanding more about multiple breeds in a species should provide information that can be used to base new breeding technologies on. So we can look at regions that are conserved between local breeds relative to production breeds. And that's a particularly also important question in low middle income countries as well, where production animals can be imported and indigenous animals are already, understanding why they're thriving in those environments is a similar question. So the more we can understand about the genetics of those animals, the better. And that having these breed specific information for local breeds does provide the tools to do that.
Okay, thanks, Emily. Somebody would like to add something, or? Okay. So there is I think we have to go for the conclusions from Mark, but this is just one last question related to big breeding. It's for all of you, or perhaps Javier a bit less, but it could be translated to aquaculture. So is your opinion today more important to breed towards animal welfare in big breeding, or do we need to work on low carbon footprint, feed conversion rate or health? Or I guess it was a bit already answered before, but yeah, Peter, Emily, Mark, if you want to comment before making the summary and say bye-bye, it will be great.
Can I come in on that? Look, I think both breeding for animal welfare and breeding for low carbon important. We can't just say we have one goal and we ignore the others. The challenge, and Javier, talked about how we all are aware of that challenge for sustainable food systems, is how we make progress on a number of goals, sometimes where there may be elements of conflict. But when you look at low carbon, study after study is showing that in the developed world, in other words, in the EU, Australia, the US, we need to eat less meat. Again, this is not talking about vegetarianism, but it's saying less meat, because the emissions from any livestock are nearly always going to be higher than from plant-based diets. And a lot of studies are now talking about adopting the need to adopt flexitarian diets. In other words, diets in which there is meat, but not large amounts 3 times a day, every day. And in that way, we can very substantially reduce the greenhouse gas emissions from our food systems. But of course, we also need, vastly improved pig welfare. Pig welfare in the EU is often very, very poor. Just to take one example, nearly all pigs in the EU are routinely tail docked, even though the ban on routine tail docking came into force 30 years ago. Thanks.
Okay, thank you very much. I think it's time for the wrap up from Mark. Thank you very much, all of you, for coming. Thank Çağla for excellent work moderating. And yep, thanks for coming. Mark, the floor is yours.
Thank you, Anna. And I think I'd just like to take this opportunity to thank Anna, and we've seen glimpses of Isabella and Çağla. Thank you very much, Çağla, for chairing that session. You've all put a lot of effort into this really useful and beneficial webinar. Yeah, I think there's been some really interesting concepts raised. I particularly liked Emily's presentation I'm going to change her title from FAANG to forked. It'll be FAANG to phenotype to fork. And I thought that was really useful. And, we're only at the early stages of this. On comparative genomes is something that Emily mentioned and will be really useful for looking at different phenotypes. Yes, I think it was an interesting discussion also by Mr. Christensen earlier on. I think I'd like some clarity on he mentioned gene editing, but he also mentioned GMOs within the European Parliament context, which was news to me. So hopefully we can get some feedback from Anna on that. That will be quite interesting. A few other notes I took on today's discussion. I think Peter raised some very interesting points, and yes, the breeding companies are fully aware of this, and I think that was reflected in the responses from the question on the major hurdle for new breeding technologies and for moving into the new era of animal breeding is public perception. And I think the breeding companies, Peter, I'm sure you're aware, are fully aware of this. And within the current genomic selection, obviously sustainability and welfare and disease traits are up there with the usual traits that you were mentioning. And I think, just let's also acknowledge the huge advances that genomic selection has done over the last 20 or 30 years. And we can hopefully use this to improve animal welfare and disease resistance with conventional genomic selection. Thank you, Javier, also for the interesting insight into aquaculture and the importance of the sustainability development goals there. And I think there were some other very interesting questions raised by the various people on the webinar. And I think the point that was put across by a number of people was that we need to have, and obviously a lot of breeding companies already do so, is have a balanced breeding program with welfare as a major factor. So yes, on that note, I think we hope that people will finally look towards this as something positive, and we hope that what's next in animal breeding will lead us to a more environmentally sustainable production system with more disease resistance. That's also an advantage to both the consumer and the animal itself. So with that, I'd like to thank the speakers for their interesting points of view and their data. And hopefully we'll see you on the next webinar, which I think Anna will probably circulate details of when that will be. Thank you, Anna.
Yes, thank you. I think, Mark, you had a question for me, but I was writing in the chat to Javier. He had to leave before the end, so I'm sorry I didn't got your question. So if you want to repeat it, it will be great. No, you hadn't a question? Okay, that's you were asking me to comment something, and I didn't. I was writing, so I didn't.
Oh no, so I was going to say that you'll update everyone as to when the next webinar series will be. When the next webinar.
Okay, sorry. That's I thought before. Yeah. Okay. So yes, thank you very much, everyone, for coming. Thanks to all our speakers and panelists. Thanks, Mark, for the wrap-up, and Çağla for the moderating task. Yeah, we will go back with another webinar, and in 2022, even if we prefer in-person meetings, has to say that these webinars are very interesting because we are having a lot of audience and a lot of exchange about animal breeding practice and research. So that is perfect. Çağla is telling me what it was about the comment. So yes, Asger Christensen was mentioning GMOs, and yes, that's there is no discussion about GMOs in animals at European Parliament. What is already ongoing is what is was stated by the European Commission in April 2021 this year. So, and afterwards in UK is that genome editing tools has the potential to improve sustainability, and so we have to move to make a new regulation around. It's not about deregulation, but to have another regulation, more modern regulation adapted to these new technologies. And for plants, they have started that policy action for mutagenesis as cisgenesis, what is not already the case for animals. We have to explore more safety data. We have to provide more safety data around animal breeding, new breeding techniques. So, but this is not question. I mean, this is not a question about GMOs or not. It's about new genomic techniques. That's the wording of the Commission, and it includes, of course, a number of different techniques. But the purpose is to regulate differently mutagenesis and cisgenesis at this moment for the plant sector. Further actions for animals in the future, they are taking a look at the potential of new genomic techniques in animals. But it is not discussed at the European Parliament. I mean, there are debates, but there are more about genome editing tools and not GMO products. I thought this answered to your question, and we are just 1 minute to 3:30 Central European time. So thank you very much all for coming today to engage discussion with us. We will inform you about next webinar, and we will send you very briefly a summary of today. Thanks again. Have a nice afternoon and week. And Merry Christmas, of course, but and yes, we have here Isabella, and the rest of the team has been supporting the organization and follow-up of this. Emily, thank you very much. Peter, thank you very much. Just here with us. Bye-bye.